All right, so chapter 23 feels like we're kind of skipping around in the book, but I want to make sure that we talk about indicator organisms because they are particularly important for the water environment. So what we're going to do is we're going to spend a little bit of time getting used to what an indicator organism is, and then talk about some of the tests that we use for total and fecal coliforms, because that's what's going to start to be important when we start to look at drinking water and wastewater treatment. So first things, when we talk about why do we need organisms? When we try to locate or sample for microorganisms, it can be tedious, difficult, time-consuming, and expensive. So therefore, if we can find an organism that is indicative that there is another pathogenic organism or something there in the system, that's going to save us time, energy, all that good stuff. So the way that we define an indicator organism is it's an organism whose presence indicates that pathogenic microorganisms may also be present. So when we say pathogenic means it causes disease. Now it can be human pathogenic, which means it causes diseases in humans, or it can be pathogenic to other organisms. So you can have plant pathogens that cause disease in plants. So generally when we say pathogens in this class, we're talking about things that cause disease in humans, but just be aware that they can cause disease in other organisms. So the point of an indicator organism is to be able to find whether or not we think within a reasonable measurement, basically, that we have pathogenic organisms. So they were developed specifically to look at fecal contamination. So when we say fecal, it's exactly what you think it is. It's feces. It's what's shed out of your body as excrement, right? And so unfortunately, we're going to have to talk about that topic. And basically the idea of an indicator organism is that non-pathogenic bacteria occur in the feces of all warm-blooded animals. And so if we can find some of these indicator organisms, they may tell us if there's other things going on in the system. So that's the purpose here. The other thing that I want to make sure we talk about as we start to get into this is what we call coliform bacteria. This is going to be hugely important if you go into drinking water or wastewater. Normally, coliform bacteria occur in the intestines of all warm-blooded animals and are excreted in great numbers in feces. So in all of the waste, right, that we have in wastewater, that's got feces in it, and so it's going to have coliform bacteria in it. We know from lots of studies that in polluted waters, the coliform bacteria are roughly proportional to the degree of fecal pollution. So we found this out through lots and lots of unfortunate waterborne diseases. Fecal contamination is directly corresponding to coliform bacteria presence. So generally, coliform bacteria are hardier than disease-causing bacteria. And what that will tell us is that if the coliform bacteria are absent, then most of the time the water is bacteriologically safe. Doesn't mean it's 100% safe, but it's a good indication, hence the name indicator organism. If we find coliforms present in water, that indicates that there might be other microorganisms there that have the potential to cause disease, and that water is potentially unsafe to drink. So again, we're not detecting every pathogen. That's not the point. An indicator organism tells us something about the safety relative to a standard, right? So coliform bacteria presence means it's probably unsafe. Coliform bacteria absence means it probably is safe when we think about drinking water. So this is a list of the criteria for an ideal indicator organism. So it needs to be useful for all types of water. It should be present when enteric pathogens are present. And enteric means part of your digestive system, right? So coliform pathogens. The organism has to have a long survival time so that we can use that to say that there are probably hardy enteric pathogens here as well. So we don't want an organism that's going to die off before the pathogens do. So that's important. It shouldn't grow in water. Testing should be easy to perform. The density of that organism should have some direct relationship to the degree of fecal pollution, and the organism should be a member of that intestinal microflora, so what's going on inside your gut. So these are kind of our criteria for indicator organisms. And this is just, unfortunately, a very disgusting table that tells you how much of an indicator organism is in raw sewage. So for example, here we see coliforms have anywhere in the range of, let me just get close so I can read the number, 10 10 to the 7th or 10 to the 8th CFU per 100 milliliters. That is a very, very high amount for a very, very small volume. So you can read through this and see some of the other things that are unfortunately in raw sewage. And then the other thing that we want to talk about when we talk about indicator organisms is that there's different types. So there are process indicators, fecal indicators, and what we call model organisms, and they each are slightly different. So just so you know, if these terms come up, I'm not going to quiz you on the difference between these, but just so you know that there are slightly different ones, especially if you start to get into depth when it comes to these organisms. Okay, so let's talk about what total coliforms are and why that's important to us. So the coliform group is used as the standard of assessing fecal contamination. It's been used for assessing recreational waters. It's been used for wastewaters for quite a while. And so we've learned that it's optimal to take about 100 milliliters of a drinking water sample when we're doing this type of testing for total coliforms. So what I want to point out on this particular slide is we have what is this high level of Enterobacteriaceae, which is a group of enteric bacteria, right? So in your gut, enteric, right? And the total coliforms it's a big group of that group. Like there's other things that are in that group that are not considered coliforms, but the total coliforms is kind of that headwater of all of what comes next. So fecal coliforms are a subgroup of total coliforms. These are the ones that are in feces that we know are pathogenic. And just as an example, E. coli is an example of a fecal coliform. So we can test for total coliforms, we can test for fecal coliforms, and that tells us that we're reasonably certain that E. coli is probably present. So that's kind of what's going on there. So when we talk about indicator organisms, we don't want to make it sound like they're 100% positive or 100% negative. So there's some drawbacks to using them that we have to be very aware of. So we want to be careful because we can have regrowth of organisms in aquatic systems or in our drinking water distribution systems. And so if they're regrowing, that could be a false positive in the system. We could have a suppression of the indicator organism based on high bacterial background growth. So there could be a lot of bacterial growth going on, and because of that, we're not seeing a signal from a specific indicator organism. That's an issue. They're not directly indicative of a health threat. So that's important, right? Just because we say that an indicator organism is there does not mean that it's a health threat. It means that it's likely a health threat, and that's important. And then there is no relationship between those indicator organisms and enteric protozoa and viral concentration. So we're not using indicator organisms to look for protozoa or for viruses. And that's important to be aware of, right? We can't use an indicator bacteria to say a protozoa is there. So that's important to know. Okay, so let's talk a little bit about coliforms. So total coliforms, remember, is that big group, right? Their die-off rate of that particular bacteria depends on the amount of bacteria present, the type of organic matter that's in the water, and the temperature. So if they have the organic matter that they want and the temperature that they want, they're going to grow quite well. So one thing that's important when it comes to total coliforms is that we don't want them in our drinking water. And one of the problems that we have with coliforms is they may actually colonize and grow in biofilms and distribution pipes. And when they do that, even in the presence of a disinfectant like chlorine, which we'll talk about, they can still grow. So they may be present in a biofilm and not as a cell floating around in solution. And so they may actually be present, but because we can't find them in solution, we think that they're not there. So that's important to know about these guys. E. coli, as an example, is 2,400 times more resistant to free chlorine when it's at a surface. So normally what we do with E. coli is we shock it with chlorine, and that causes the cell to lyse or break apart. But when they're in biofilms and hanging out in colonies, they're actually very resistant. So this is one of the challenges with our indicator organisms and total coliforms in particular, is that they have this colonial behavior sometimes. So just something to be aware of. All right, we're going to very quickly talk through a couple different tests specifically for total coliforms. So you saw the MPN test in a previous video. We're going to talk very quickly about presence, absence, and call alert, because these are specific tests for total coliforms, and these are the ones you are most likely to come across as an environmental engineer. So the MPN test consists of 3 steps, and most of the time we're doing the presumptive test, not the confirmed or the completed test. I will show you what each of those are, but we're most likely doing the presumptive test. So the presumptive test is what you've seen before. We take our sample, we do a specific type of dilution, we add in the chemicals that we need or into the broth that we have, we do the incubation, and we look at color change, and then based on the number of tubes that changed, we go from there. So most of the time we're doing the MPN presumptive test, which you've seen in the past. The confirming test basically we'll take one of those samples and we will use specific agar designed for total coliforms to do either a positive or negative test. So we have, for example, endo agar or this Levine's agar on the left side of the screen, and we can take the same sample, the same water sample, and we can plate it out to see if we get positive or negative results. And these specific agars confirm that MPN test. That's why it's called the confirming test. Now, I don't have a slide on the completion test, but the completion test basically takes it a step further and does both of them and compares the two. So I'm not going to talk too much about that. I want to make sure that we talk about the membrane filter test because we talked about membrane filters when we talked about water sampling, and it's easier to perform because there's fewer test tubes and less labor than MPN. So normally we'll measure about 100 milliliters of drinking water through a membrane filter with that pore size of 0.45 microns and trap the bacteria on the membrane surface. And then the membrane gets placed on that agar to make sure that we permit growth. Now, if we're specifically trying to get to total coliform growth, we want to make sure that we use something like an endo broth media specifically. So we've seen membrane filter before, same idea here. We just, if we're looking for total coliform specifically, we need a specific type of agar. So here's that membrane filter test again, just as a refresher. Remember that you're taking your sample, you're applying vacuum, and you're pulling that water through the pad. And then the pad is either put directly onto the agar or it's dissolved in saline before it's put on that agar plate. The last test that I want to make sure that we briefly hit before we finish up this pre-lecture is the presence-absence test. And this is a qualitative, not a quantitative method. So all it does is tell you whether they're present or absent. It doesn't tell you how many are there. So we use a very specific type of broth that is also used in the MPN test, but we just don't do the dilutions. And that's basically the presence-absence test, which is really, really easy. But in recent years, in terms of presence-absence tests, we use what are called enzymatic assays, and they're allowed to, what they do is they allow us to detect total coliform bacteria and E. coli in drinking water in the same test. So what we can do is we can basically expand that PAA test with a couple more chemicals in order to make sure that we understand the presence of E. coli and total coliforms. So this is a long procedure, so you can feel free to pause the slide, but this is an example of an MPN PA assay. It's an enzymatic assay called ColAlert, and we're actually going to do this in lab. So ColAlert, basically what happens is we take our water sample and we add specific enzymes to it. And those enzymes create specific reactions, and those reactions are slightly different for total coliforms and slightly different for E. coli. And what that allows us to do is it allows us to count by like a presence-absence test how many are positive for total coliform, and then E. coli specifically will fluoresce under UV light. So we will actually look at samples under UV light, and we'll be able to see that when we have the fluorescing samples, that's going to tell us something about E. coli specifically. So just take a pause, read through this procedure. We're going to perform MPN, presence-absence, ColAlert, all in lab, and we're going to use membrane filters as well. So we have a lot of different tests that we're actually going to try out in class related to these indicator organisms. So to kind of bring this all back together, the reason that we care about the indicator organisms is because things like E. coli and total coliforms are going to tell us that other pathogenic bacteria might be there. Now, we don't want E. coli, right? That's not a good thing to have, but it's also going to be indicative that there might be other bacteria in the water that we don't want to be drinking. So that's the importance of doing all of these different tests.